So today's DNA metabolism, which is what, really? So what is DNA metabolism, right? It's basically, it provides storage for our genetic material and information. So the structure should be all the, the whole point of it is to store memory, so it should always remain the same, right? So, but it's not so static, right? It's a chemical, so it's going to degrade naturally with a certain rate. So there's enzymes that'll fix that damage. And then every time a cell divides, we have to make a copy of it. And that copy has to be of high fidelity. What does that mean?
No errors.
Right, or few errors. It's impossible to have really no errors, but we want to limit those errors to as few as possible. So errors can arise during and after that DNA synthesis, and they're constantly checked for. Repairs are made. Segments of DNA are rearranged either within a chromosome or between two DNA molecules. This is called recombination. Have you guys ever heard of this concept? Where DNA were recombined. And it's almost holiday time, so we're going to talk about the holiday junction, right? I guess summer's holiday, right? Normally it makes more sense when I present this around Thanksgiving. So that's recombination. We'll look at the holiday junction. And then DNA metabolism consists of a set of tightly regulated processes that achieve these tasks. And there's three fundamental rules of replication. Replication is semi-conservative. What does that mean?
One parent strand.
Yeah, exactly. So one parent strand in the new strand and one daughter strand, one new strand. Replication begins at the origin and will proceed bidirectionally. So there's an origin of replication typically, right? And then it proceeds bidirectionally, meaning both ways away from that origin. Synthesis of the new DNA will occur in the 5' to 3' direction and is semi-discontinuous. What does that mean? Semi-discontinuous. One of the strands is continuously produced, the other one's in chunks. And you know what that's called? Those pieces? Yes, the Okazaki fragments. See, I don't need to cover this chapter. You know the chapter. I skipped the other 2 chapters for no reason. So, DNA replication is semi-conservative. You guys ever seen this experiment where they grow the bugs with the heavy nitrogen? Nothing but heavy nitrogen, and you can separate that out on a density gradient, and you see where the DNA, the chromosomes end up. And then at one point you switch to regular nitrogen, regular N14 nitrogen, and you have these cells timed so you know exactly what the generation time is, so you can take them out after one generation. You see that it's halfway between where you would see the N15 only DNA and the N15 N14. It's a hybrid, and as you keep going, you generate more and more just N14, and you keep the one hybrid set of DNA. So that's how they proved that basically it's semi-conservative, because if this was not semi-conservative, if this was, what would you call it? Just conservative. The parent strands, you'd see one band here for the N15 and then one band for the N14. But they were able to prove that it's semi-conservative, one daughter strand and one parent strand based, because you have this intermediate band where the two, the parent strand is containing all N15, the daughter strand all N14. So I don't know if we need to go through that because we just described it. So, and I just described this too, didn't I? So we can skip these slides. Review of semi-conservative replication from chapter 8. And again, one parent strand and one daughter strand. One parent strand stays with the new daughter strand. Bidirectional replication force. Once replication begins, it proceeds at the same or opposite direction. Carnes experiment, the DNA was radiolabeled with tritium DNA and was isolated, spread under a photographic emulsion. So the tritium will cause that photographic emulsion to allow you to see where the tritium is. And it shows circular DNAs with an extra loop. It showed that both strands are replicated simultaneously, and the two replication forks is bidirectional. Oh, they don't have a picture of this one, do they? No. I should get that from my old PowerPoint. There's a picture where they grow it on tritium, and then they cut back the amount of tritium, and you can really see the picture itself with the bidirectional fork. But oh well. And it proceeds in a 5' to 3' direction. So the leading strand is made continuously. We already discussed the lagging strand is made in fragments, and that's because it has to be made in this 3' to 5', so it has to constantly starting new fragments at this replication fork, whereas the leading strand can continuously be made 5' to 3'. So, what is the chemistry? Do you guys know what the chemistry? How do these new bases get added to the strand? So both of you guys took methods too. You didn't. When we sequence DNA, what is it that we add that generates short strands, and they're the terminator bases? What is it about those terminator bases that allows them to stop the DNA polymerase reaction when that base gets incorporated? Do you guys remember? I was going to quiz you today. It's not a particular enzyme, it's a particular chemical modification to the base. So you need the nucleotide triphosphates to add bases, right? But how do you stop those bases from adding? What is it special about those terminator bases? That they stick the dye on so you can see them? Do you remember? Remember they're the di-deoxynucleotide triphosphates? So they lack two OH groups, the 2' and the 3'. So there's no 3'. So this is the base, right? Normally you're going to have a 3' hydroxy group, right? And the dideoxy lacks that, so there's no place to add the nucleotide. So what happens is this 3' hydroxy attacks that first phosphate in the nucleotide triphosphate, and I got a picture right here. So this 3' hydroxy group attacks the phosphate in the 3', the nucleotide, the incoming nucleotide triphosphate, and it just does nucleophilic attack like that, and then you release this pyrophosphate as the result. So, and then this is added a new base, and you have a new 3' hydroxy group to do the next attack for the next base coming in. And required for this is magnesium. If you guys remember when you set up PCR, I think in methods 1, you had to optimize your PCR reaction, and one of the ways was alter that magnesium concentration. And then there's a couple of aspartic acids that help coordinate that magnesium along with the binding the nucleotide triphosphates. DNA polymerase requires a primer, right? So we have to have some short nucleic acid sequence that binds to the template strand that allows for a 3' hydroxy group to add a new base onto. Now this primer can be RNA or DNA. In PCR, we normally use a DNA primer, but for normal DNA replication, it's going to be an RNA primer that the cell creates via an RNA polymerase. That gets added first, and the enzyme is called primase that adds this primer. So Some features of DNA polymerase: there's an insertion site where the incoming nucleotide binds, and a post-insertion site where the newly made base pair resides when the polymerase moves forward. And then there's, you can think of it, everybody says it looks kind of like a hand kind of wrapped around, where you have the thumb and the palm, and then the insertion site and the post-insertion site. And then it just moves right along. And there's a proofreading activity that this has. So DNA polymerase adds nucleotides or can dissociate. The number of nucleotides added before dissociation is called the processivity. Now there's different DNA polymerases in the cell depending if it's eukaryote or prokaryote. They all have different DNA polymerases. The one with the highest processivity is the one that generally is used to replicate the whole genome. You have lower processivity DNA polymerases also, and those are typically involved in DNA repair or sealing up those, getting rid of the RNA primers and the Okazaki fragments and just getting rid of those and replacing it with DNA. So polymerase has wide varying processivity rates. The error rate is very small. So errors in E. coli are about 1 in, 1 billion? 10 to the 9th? Yeah, 1 billion. To 1 in 10 billion, or about 1 per 1,000 to 10,000 replications, you'll add one base add. Why are we adding wrong bases? Do you know? Why can you get wrong bases added? Remember keto-enol shifts, tautomeric shifts? That can occur, you get those tautomeric shifts in the bases also. So you can, get a tautomeric shift and accidentally insert an incorrect base. Does this show this up here? Let's see. Now this just shows you where incorrect base pairs don't really fit into the pocket. So that's one way that it reduces the amount. But you will get incorrect bases inserted when you have tautomeric shifts through those keto-enol tautomers. You guys remember that? And guanine can exist in this alcohol, the enol form or the keto form, and just like the enol form of thymine versus the keto form of thymine. And then it'll base pair with the wrong base, if temporarily. But these keto-enol shifts are very transient. They're heavily favored in one direction. So you'll get a shift perhaps while it's being incorporated, and then shortly thereafter it'll shift back and it'll show that it has an improper base that's been added. So errors during synthesis are corrected by DNA polymerase has 3 to 5 prime exonuclease activity. So we add 5 to 3 prime. If an error gets inserted, it can just it'll stall if you insert the wrong base. The next base will stall, and then gives time for this 3 to 5 prime exonuclease activity to occur, and then the 3 to 5 prime exonuclease just cuts out the wrong base and then allows a new base to come in. So it's kind of like its proofreading ability, that 3 to 5 prime. So. There's at least 5 DNA polymerases in E. coli. There's DNA polymerase 1, DNA polymerase 3, and then there's 2, 4, and 5. And they're all involved in DNA repair. DNA polymerase run is very abundant, but it's not ideal for replication. It has extremely low processivity. Its rate is about 600 nucleotides per minute, slower than that observed for replication fork movement. Its primary function is just to clean up, get rid of those RNA primers and whatnot. Polymerase 3 is the principal replication polymerase. It has super high processivity and can stick around for a while. And then there's 2, 4, and 5, which are involved in DNA repair. Polymerase 1 also has 5 to, not just 3 to 5 prime exonuclease activity, it has 5 to 3 prime exonuclease activity. So it can chew bases up as it adds them, right? And this is called Nick translation, if you ever heard of that. You can actually use that in a lab to introduce labeled bases if there's some, you want to label a plasmid or something. You put a nick in there, or you're going to have a nick in your DNA. You add complete pol 1 with the labeled bases you want to add, and it'll add bases by a process called Nick translation, because it chews off from the 5' end as it adds a new base to that 3' end. And it still has the 3 to 5' activity, nuclease activity. It moves ahead of the enzyme, hydrolyzes things in its path. It's known as Nick translation. A strand break moves along with the enzyme, and this activity and the polymerase activity are in the Klenow fragment. This gentleman, Klenow, discovered if you treat polymerase 1 with a protease, it separates into 2 fragments. The Klenow fragment has the 5 to 3 polymerase activity, the 5 to 3 prime nuclease activity and the polymerase activity. A distinct domain that can be separated by the protease treatment, which I just said, because I don't need to read the slide. So DNA polymerase 3 is complex structure with at least 10 types of subunits. There's 2 core domains, alpha, epsilon, and theta. And the core domains are linked by the clamp loader complex, which is this tau 2 gamma delta delta prime. The core domains each interact with a dimer of beta subunits that increases the processivity of the complex. So there's a clamp loader, there's these beta subunits that kind of clamps it to the DNA, which increases the processivity enough where you can get the complete genome to be copied. It forms a sliding clamp that prevents dissociation and gives you processivity greater than 500,000 base pairs. And this is kind of like what pol 3 looks like. Here you have a sliding clamp, pol 3 core, and then the clamp loader, which loads it onto the DNA, and then the sliding clamp, beta sliding clamp again. That one's pointing to the wrong thing, huh? This is the clamp. And it looks sort of like this with DNA in the middle. That sliding clamp does, anyway. And this is just a table from your book showing you all the different subunits. So it requires over 20 enzymes and proteins for DNA replication. And the whole set together is known as a replisome, because it replicates the chromosome. There's helicases, right? Remember, to copy the DNA, you have to unwind the DNA and melt it into single strands. So helicases add that energy to separate those 2 DNA strands. We can do that in the lab by heating it up, so we add energy to separate them that way. Helicases use ATP to unwind and separate those strands, because you have to separate the strands before you can use one as a template, right? Topoisomerases, they'll relieve the stress caused by separating those strands. You generate the supercoils, front and back when you pull apart those strands. So you're going to need topoisomerases to relieve that stress. Then you have DNA binding proteins. So once you separate those strands into single-stranded proteins, or not proteins, single-stranded nucleic acids, those single-stranded nucleic acids aren't happy to be in solution like that all by themselves. They like to base pair. So they're going to want to either base pair with themselves or base pair back to the template. So we use DNA binding proteins, or single-stranded binding, SSBs. I think the last slide, this. These whole C's interact with SSBs. These are single-stranded binding proteins, is what that SSB stands for. And they basically stabilize those single-stranded DNAs. Primases are enzymes. It's an RNA polymerase that makes that RNA primer, so the DNA polymerase has a 3' hydroxy group to add sequence to. And then there's DNA ligases, which are required to seal the nicks. You have a lot of nicks after, that lagging strand due to all the Okazaki fragments. So you're going to need a DNA ligase, which goes in there and seals that nick. So initiation of replication in E. coli begins at the origin of replication, ORIC. So anytime you have a plasmid that you want to grow in E. coli or some bacteria, it's going to have an origin of replication. Sorry, I can't. Nut dust in my throat. So its highly conserved sequence contains 5 repeats of a 9 base pair sequence, the R site that forms binding site for the initiator protein, DNA A, and it contains a AT-rich DNA unwinding element, or DNA unwinding element. Why would that unwinding element have a lot of AT groups? Why not just CGs? Base pairs, or not base pairs, yeah, hydrogen bonds. God, I can't think. The base pairing between A's and T's only has 2 hydrogen bonds versus 3 between C's and G's. So it's a site that's easier to unwind, right? DNA A has I sites, the IHF is the integration host factor, and then the FIS, factor for inversion stimulation. So there's your do sequence, all these other sites, and then the 5-9 base pair sequence consensus is TTAAT, NCACC, and so forth. So all those different sites we find at the origin. DNA A proteins are ATPases, so they're going to burn ATP, and 8 DNA A's bind the R and I site. DNA wraps around the complex, forming a positive supercoil. That positive supercoil puts strain on the double helix, which helps it to unwind and denature at the do region, where you have all the A's. And then strain leads to denaturation. Associated proteins then facilitate the DNA binding, and you get DNA C, ATP, and they'll stabilize that, little spot that's unwound. DNA B hexamer structures opened by the binding of DNA C, and then DNA B will bind to separate strands. Where do they have that? DNA B migrates along the single-stranded DNA in a 5 to 3' direction and unwinds the helix. Other proteins, such as POL3, link to the DNA B, and then the single-stranded DNA protein, single-stranded binding proteins will stabilize those separated strands. You have to have the single-stranded binding proteins, and then DNA gyrase will relieve the stress ahead of those replication forks as that starts to begin to move. So initiation is regulated to occur only once per cell cycle, right? You don't want to make multiple copies of chromosomes. It takes a lot of energy and a lot of time to copy the whole genome. You only want to do it one time when you're going to divide the cell once, right? HDA binds to the beta subunits of DNA polymerase 3 and stimulates hydrolysis of DNA A's ATP. HDA is known as homologous to DNA A. Simple names for these: HDA, homologous to DNA A, because it's similar to DNA A. DNA A complex then associates, ADP dissociates, ATP rebinds to DNA A to stimulate all over again, and the time scale is about 20 to 40 minutes in E. coli, which is about their, generation time, right? So regulation of replication initiation via methylation. So if you guys remember from micro class, or if you had it, microbes tend to methylate their DNA, right? So there's this, after replication, the origin is hemimethylated by DAM methylase. DAM Methylase means DNA adenine methylase, methylates the N6 of A and GATC sequences. The hemimethylated origin sequence interacts with the plasma membrane uses seq A, and after a period, the seq A dissociates. Origin C sequences are released from the membrane. The DAM methylase fully methylates the DNA to allow the new DNA to bind. So once you replicate, it's just hemimethylated. So what importance would that have, do you think? Having a hemimethylated replicated chromosome rather than, why doesn't it just methylate it as soon as it makes that sequence? As soon as it copies it, why does it wait? Any ideas?
Needs to stick to the side of the cell.
Perhaps that helps with that, but the real reason is for error. If you introduce errors during replication, which is the time where you're going to get a lot of errors introduced, if you're going to have any, the parent strand can be identified because it's going to be methylated already. The daughter strand won't be methylated yet, right? So you'll have you'll be able there'll be enzymes which will be able to recognize which sequence has the original sequence and which one has the error in it, so it knows which side to correct. Does that make sense? So, second phase of replication is elongation. DNA primase, DNA G, makes RNA primer. Primase interacts with DNA B helicase, but the primase moves in the opposite direction to the helicase. DNA polymerase 3 adds nucleotides. Then pol 3 is linked to the DNA B, which is tethered opposite the DNA strand. And as in leading strand synthesis, primase makes the RNA primer and DNA pol adds nucleotides on the lagging strand. But the same DNA pol works in both strands. The DNA lagging strand has to loop around, right? Have you guys seen those types of pictures? You don't see them here, but like this. The leading strand can just follow, and that as that replication bubble gets pushed along in that 5 to 3 prime direction, the leading strand just can just keep going. But for the lagging strand, it has to be looped around, and you add new DNA polymerase as it loops around continuously, right? Because it's adding strands. It's going to add from this direction back, and then so it can't go with the bubble. You kind of have to keep adding DNA polymerase complexes onto that. And this shows you the lagging strand synthesis also. Hold on, I got it. Expecting a delivery. Something about credit cards, card holder services. Huh? My air conditioner's shot, so I'm getting a freight delivery to my house. Waiting for that. Sorry. So, this just shows you how the lagging strand works. Any questions about this? And it has to keep going. The old one will get left behind. You get a new primer made as the clamp loader loads a new clamp on after the primase makes that new primer. And it goes on and on continuously making that.
So on the leading strand, put a primer on there and it just kind of...
It just keeps going.
Link.
Yeah.
On the lagging strand, they put a primer and they kind of work backwards and then
Yep.
Trying to run work backwards to the last one.
Exactly. Continuously, you have to make all these little fragments, and then eventually those RNA primers will have to be removed. DNA polymerase 1 does that with its 5' exonuclease activity, 5 to 3' exonuclease activity. Chews up those DNA primers and replaces them with DNA bases rather than RNA bases. And then the nicks will eventually be sealed via DNA ligase. And you'll have a complete piece of DNA there that's copied. So.
When you say nicks, it means all the base pairs are there, they're just not connected at one spot?
In one spot. And it's just a single nick. It's not like it's all the bases that are there unattached, but you're going to have a nick where the DNA polymerase, when the pol 3 makes it to the next primer, it's going to stop and it won't attach that piece of DNA to the primer, because that primer doesn't have, it's not a trinucleotide. Well, I guess it would be a trinucleotide, but it doesn't have the ability to attach that. So it leaves that there. And you'll have a nick. They don't show you a picture where the strands come together, do they? No. Well, yeah, right here. So here you're going to have the pol 3 is going to stop, and there'll be a nick there. So eventually, as this keeps going on, there'll be a place where DNA pol 1 can come in on this nick and do what's called nick translation, where 5' to 3' exonuclease activity chews up that RNA primer and replaces it with DNA. And where it goes on, there'll still be a nick, right? But it just moves along down the thing and keeps replacing down the strand of DNA, keeps replacing the bases with new DNA bases. But remember, pol 1 has very low processivity, so eventually it'll just pop off. And then there'll be a nick where it pops off, where it'll be DNA and DNA. DNA ligase will come up behind it and just attach those back together. Do they show you that? Transitioning between Okazaki fragments. The core subunits of DNA pol 3 will dissociate from one beta clamp, bind to a new one. The RNA primers removed, polymerase 1 will fill in the gap, removing that primer. DNA ligase will then come in and seal that nick. So I guess I could have skipped to this slide, because here it shows you that nick. DNA polymerase 1 comes in, chews up the RNA primer, and it goes until it disassociates. It has low processivity, so it's not going to go for more than like 90 bases or so, and leaves a nick. So then DNA ligase will come in and seal that nick with a simple reaction. So how does DNA ligase work? The 5' hydroxy group must be activated by attachment of an AMP. 3' hydroxy acts as a nucleophile and attacks that phosphate, displacing the AMP. So here you have AMP, the enzyme DNA ligase. ATP comes in and forms, it basically adenylates, the amine group on the DNA ligase. So you have an AMP then, so it's like an activated AMP. Can then adenylate that, comes in and adenylates that phosphate that's left over on the right here on the 5' end. Here's, you have your 3' hydroxy group, and then this is activated because you have an ADP now there attached. This 3' group attacks the phosphate that was originally there, and this AMP makes a great leaving group, better than the water that would have left before, or hydroxide that would have left before, if you didn't have the AMP added to that, and basically seals that nick. So you have the 3' to 5' phosphate there, sealing the nick. Third stage of replication is termination. Replication forks meet at a region with 20 base pair sequence called the termination sequence, TER. TERA through TERF. TER sites are found near each other but in opposite directions. Creates a site that the replication forks cannot leave. TER is also a binding site for the protein TUS. which stands for termination utilization sequence. Causes a replication fork to stop. So you get the origin, starts here. Remember, E. coli has circular DNA chromosomes. Gets to the tersites, and it forms the fork trap and terminates the DNA sequence. In eukaryotes, it's a little more complex than in bacteria, because remember, we have chromosomes that aren't circular. So we have, yeast has 400 well-defined origins, so instead of having just one origin in replication, we have multiple. And they're called autonomously replicating sequences, ARS, or replicators. The entire genome's replicated during one time per cycle. Regulation is due to the cyclin proteins, which are regulated by cyclin-dependent kinases. Have you guys ever heard of CDKs? There's often mutations in these that can cause cancer, because the cyclin-dependent kinases regulate cyclins. Cyclins regulate the cell cycle, basically, in replication. Cyclins are ubiquitinated for proteolytic destruction at the end of mitosis phase. So, and have we talked about ubiquitin? I think that's later on, right? So initiation of replication requires a replicative complex. It's the origin recognition complex, the ORC, loads a helicase onto the DNA. The ORC functions like bacterial DNA A. The helicase is a hexamer of many chromosome maintenance proteins 2 through 7. And MCM 2 through 7 functions like the bacterial DNA B helicase. So it helps to unwind that DNA. And this shows you the ORC at the origin. The CDC6 comes in, requires bound to ATP. MCM 2 through 7 come in. ATP gets cleaved. This comes off then. And then still have it bound to ATP, and then the helicase, it kind of bends that DNA, and you're going to be able to unwind the DNA there and start that complex. Eukaryotic replication occurs slowly, but from many different origins, so that way you have many complexes copying that DNA at the same time, so we can compensate for the slow replication process by multiple origins every 30 to 300,000 base pairs, so we can still get the job done. And then rather than DNA pol 1, 2, or 3, we have DNA polymerase alpha, delta, epsilon, and so forth. There's DNA polymerase alpha is probably used to make the primers for the Okazaki fragments, has primase, but not 3 to 5 prime proofreading ability. So this is a place to introduce errors, but it's introducing errors in the RNA primers, right? DNA polymerase delta is used in leading in Okazaki synthesis, so this is analogous basically to the DNA polymerase 3 in bacteria, right? Oh, and that's what it says there. And then has 3 to 5 prime proofreading ability. So if you insert a keto-enol base and then it recognizes it that it was the wrong base that was added, it'll stop and cut that base out and then try again. DNA polymerase epsilon is used in DNA repair, similar to the DNA polymerase 2, 4, and 5 in bacteria. So for DNA repair and mutations, they're going to happen spontaneously because DNA is nothing but a chemical, right? So it's subject to the same chemical degradation that just natural chemicals are. So chemical reactions and physical processes will constantly damage that DNA genome. Majority are corrected using the undamaged strand as a template. So that's why we have 2 strands. One strand can always serve as a backup copy for the other if you have damage to one strand. Some base changes will escape repair, and an incorrect base serves as a template for replication. That's how we introduce mutations, and those get passed on. If that mutation is favored, if it's a good mutation, which the majority of them are not, that the organism carrying that mutation might have a evolutionary advantage. So be more likely to have children and pass that on. But this is only if those mutations are in the germ tissue, not just, somatic tissue. The daughter DNA carries a change sequence in both strands. Accumulation of mutations in eukaryotic cells is strongly correlated with cancer. So if we just start introducing mutations with every cycle, eventually we'll accumulate mutations. Once we accumulate mutations, eventually those mutations can interfere with cell cycle sequences or proteins, and then that can result in cancer. Thousands of lesions a day, but only 1 in 1,000th will become a mutation because of the DNA repair. Human genome contains genes for greater than 130 repair proteins. Some vocabulary: a lesion in the DNA is damage, right? That makes sense. If unrepaired, lesions can become mutations. Mutations are basically where we have a substitution of one base for another. Or additions or deletions can also be mutations. A lot of mutations might be silent. If it's not in a we learned the other day that the vast majority of our sequences, they don't know why it's there for. So if a mutation is introduced there, it might be silent, not have any effect. Other times there's silent mutations if it's coding. We saw how the amino acids that are coded for by the DNA, they're what's that word? There's multiple different sequences that code for the same amino acid. If you switch one base and it codes for the same amino acid, it's also known as a silent mutation. And it's not going to really affect that function at all, right? There's types of DNA damage. Mismatches arise from occasional incorporation of the incorrect nucleotide. Abnormal bases can arise from deamination, chemical alkylation, or exposure to free radicals. So just chemical degradation, deamination, things like that. Pyrimidine dimers will form when the DNA is exposed to UV. It'll excite those pi electrons and they'll form a dimer, like a cyclobutanol between the two pyrimidines that are next to each other. So that'll cause a misshape of the DNA, and those dimers won't base pair properly either for replication. So that's a damage also. Then you can have backbone lesions that occur from exposure to ionizing radiation, such as gamma rays, or alpha or beta particles, or free radicals generated from normal metabolism. And this shows you the list of different types of damage and the enzymes or proteins that will be involved in that repair. So how do repair enzymes know which strand is the correct one? So we talked about this already a little bit. In E. coli, the parent strand is methylated, right? That damn methylase inserts methyl groups at adenines at the GATC sequences. Following a short period of time, the daughter strand is then methylated. So this time period gives the repair enzymes time to identify mismatch bases that have been wrongly inserted, and gives that time to correct those. So methylation of the DNA in repair, the newly synthesized strands unmethylated for a short period after synthesis. Any replication errors must reside in the unmethylated side. So here we have replication. The daughter strand doesn't have the methyl groups, whereas the parent strand does have methylation. So you have hemimethylated DNA. Deamethylase, after a few minutes, deamethylase will come in and methylate the new strand. And if there weren't any errors, it's both strands can then act as a template, and there's no errors there. If you have a mismatch, you'll have this little bubble which will misshape in the DNA, and these mut-S, mut-L complexes will recognize that mismatch pair and bind to the mismatch. Mut-L, mut-S proteins recognize the methylated GATC. Mut-H binds to the mut-L, mut-S DNA complex, making a DNA loop. So you see here we have the mut-L, mut-S complex. Mut-H then will bind to that, making this loop. It works its way down till it finds the methyl groups. DNA strands thread through the complex until the methylated GATC is encountered. The mismatch could be 1,000 base pairs away. But then once it does this, it cleaves the unmethylated strand. So you see it works, it binds to the mismatch. Mut H comes in and it works its way down till it gets to the GATC methylated sites. It sees which strand is methylated basically and cleaves the strand that doesn't have the methyl group added. And then Mut H cleaves the non-methylated DNA strand on the 5' side of the G. DNA will unwind and is degraded by 3 to 5' exonuclease, and then helicase 2, known as UVRD helicase, and exonucleases will work to degrade the non-methylated DNA toward the mismatch. So where do they have that? So here it degrades the DNA towards that mismatch, in this direction rather. Single-stranded binding proteins will come in and stabilize this single strand, and then you can have DNA polymerase 3 can come in and rebuild that short strand of DNA. And that's how mismatch repair occurs in bacteria. And it's those methylated groups on the parent strand, the hemimethylated chromosome, identifies which strand has the correct base. Should be the parent strand because it wasn't newly incorporated. That's the way that organism had been. There's also base excision repair, and that uses specific DNA glycosylases. Glycosylases recognize specific lesions, and they'll cleave the glycosyl bond between the sugar and the base, so it leaves an AP site, or apuronic or apyrimidic site. It's basically just the sugar there without the base. And then an example of DNA glycosylases is uracil glycosylase. Removes uracil from DNA. So what the heck's the uracil doing in DNA? No, because that would be chewed up by the DNA pol 1. There should never be uracil added, not even by mistake, I guess it could be by mistake, into the DNA. But uracil, yeah, C will spontaneously deaminate. You lose the amine group from the C, the cytidine, and it spontaneously converts to uracil. Uracil is recognized not to belong in DNA, so uracil glycosylase will recognize any uracils in the DNA and cleave the uracil out and leave that AP site. Deamination is about 100 times faster in single-stranded DNA than it is in double-stranded, right? Does that make sense? Because for double-stranded DNA, that amine group is pretty much occupied forming those hydrogen bonds. It's not going to have a lot of, and they're stacked against each other, so there's not a lot of space for water to get in there and replace that amine group, right? So other glycosylases make AP sites at 8 hydroxy Gs, hypoxanthine, and 3-methyladenine, etc., other damaged bases. But uracil glycosylases recognizes the deaminated Cs. So repair at the AP sites in bacteria is the entire nucleotides ultimately removed, not just the damaged base. In fact, sometimes the region around the AP site is removed. So a lot of times you'll remove a big chunk of the DNA around that AP site. AP endonuclease cuts the DNA backbone around the AP site and removes the DNA. So it's the endonuclease that does this, AP endonucleases. Remember, endonucleases will cleave within sequences. Exonucleases cleave at the ends of sequences. DNA pol 1 synthesizes new DNA, and then DNA ligase will come in and seal that nick again. I think they show you the base excision repair here. So here, DNA so in this case, do we see a U up there? Yeah, up there. I'm not going to jump. She told me I should use this laser. However, does it work? Ah, wow. I can do this. My old biochem teacher had a big stick when I was at UF. So here's the, can you see the U? I can't see the laser. This doesn't work anymore. You saw it working a second ago, didn't you?
Yeah.
Oh, well. You see it up there. Uracil glycosylase just cuts the U off, separates it from the sugar backbone. And then you have this AP site. AP endonucleases will come in and cleave where the damaged site is. Some of these will cleave real close, others will cleave far away, and you'll get this big chunk of DNA that gets cut out. Polymerase 1 can come in and do nick translation and replace all, where the U was with the C, and you get new DNA put in. Leaves a nick when it comes off. DNA ligase seals that nick afterwards. So large distortions in DNA are repaired by nucleotide excision, and these lesions can include pyrimidine dimers formed from UV light, 6,4 photoproducts from UV light, and then benzopyreneguanine from cigarette smoke. Pathway involves removal of DNA segment by excinucleases. So excinucleases cleave the DNA backbone in two places. You have ABC excinuclease, contains UVRA, UVRB, and UVRC. So they all have UVR because they're there to repair that UV damage. Hydrolyzes the 5th bond on the 3' site of the lesion, the 8th bond on the 5' site lesion, and removes 12 to 13 nucleotides. DNA pol 1 and DNA ligase are then, ligase replace the DNA and seal the gap again. So it makes a big gap. DNA pol 1 comes in and replaces that. DNA ligase seals the gap. They're not the gap, the what's it called? The nick. Nucleotide excision repair in eukaryotes. So hydrolyzes 6th bond on the 3' side and the 22nd bond on the 5' side. So it removes like a 27 to 29 nucleotides. And then in humans, the gap is filled by DNA polymerase epsilon, and then DNA ligase seals that nick again. And this shows you in bacteria and humans. So here you have a pyrimidine dimer that's formed. In humans, the excinuclease makes the two nicks on a much larger portion of the DNA rather than the excinuclease in E. coli. This is a 13-mer that gets cut out from, DNA helicase comes in and separates those two strands. A 13-mer in E. coli and a 29-mer in humans. Polymerase 1 fills in the gap, leaves a nick, or polymerase epsilon in humans, and then ligase seals that nick. There's also ways that you can get direct repair, and that's via photolyases. Use the light energy to repair pyrimidine dimers directly without excision. Enzymes are not found in humans and placental mammals, but it does occur. O-methylguanine DNA methyltransferase repairs the methylated guanine, and then ALK-B demethylates 1-methyladenine and 3-methylcytosine. And you have this little process here. So light, a blue light photon is absorbed by the NTHF polygu. This excitation energy then passes to an FAD. So you get electron, it gets picked up by the FAD. And then that excited flavin FAD donates the electron to the pyrimidine dimer to generate an unstable dimer. So you add the electron here, there's the dimer. The electron rearrangement restores monomeric pyrimidines. Electron is then transferred back to the flavin to regenerate the FADH negative. And then the monomeric pyrimidines are repaired. So it's in other organisms, not humans, they can just repair the pyrimidine dimer directly via absorption of blue light rather than ultraviolet light, just a lower wavelength. So how O-methylguanine leads to the mutation? So here you have O-methylguanine, it base pairs with thymine rather than with cytosine. So that's how you can introduce base pairs with the O-methylguanine. And you can have direct repair of acylated bases by AlK-B. And the methyl group, alpha-ketoglutarate, AlK-B comes in with iron, transfers that methyl group from alpha-ketoglutarate and makes succinate with carbon dioxide with oxygen in the process. Basically, what it does is you add the hydroxide group, so hydroxylates it, and then this can be removed as formaldehyde and you've regenerated the adenine. Same thing, similar process occurs with 3-methylcytosine also. So what happens when there's no undamaged DNA to use as a template for repair? So sometimes both strands are damaged, right? No template with DSB cross-links are damaged in a single-stranded DNA. Unrepaired lesion can cause replication to stall, and then you're going to repair using another chromosome as its template, which is a safe way to retain some functional sequence, homologous recombination, or error-prone translesion synthesis, which is where it just hooks lesions back up together. So here you have an unrepaired lesion, causes that single-stranded DNA, recombination DNA of the error-prone, or if you have an unrepaired break, double-stranded break, you can get recombinational DNA repair, which gives you some sort of template to repair that chromosome. TLS is part of the SOS response. So this response when DNA damage is extensive. Proteins include UVRA, UVRB, UMC, and UMD. UMU is unmutable because it lacks UMU genes, abolishes that pathway. Cleave UMD prime and UMC bind to RecA to create DNA pol 5. And this polymerase can process past the damage, but it's going to introduce errors there, right? Because there's basically a lesion with no template. It's going to kill some cells, mutate DNA in others, but some cells eventually will survive if it's not like could introduce silent mutations, or if it doesn't do too much damage, it's going to be mutations that will allow that cell to survive. So TLS polymerase is abound in mammals. Most recognize specific types of damage and have appropriate response. DNA pol eta assists with TT dimer and halts the replication fork. Most TLS polymerases are limited to short regions of DNAs, which minimizes that mutagenic potential, because it's just looking at the damage, trying to repair that, because it introduces mutations, basically. But if you can, recombination is better because you have another chromosome that can serve as a template with good sequences. Segments, they can rearrange the location within a chromosome from one chromosome to another. Such recombination is involved in many biological processes, including the repair, segregation of chromosomes during meiosis, because that will enhance genetic diversity. In sexually reproducing organisms, recombination and mutations are two driving forces in evolution. Recombination of co-infecting viral genomes may enhance the virulence and provide resistance to antiviral drugs also. So, there's a competitive advantage for this recombination. Classes of recombination include homologous general recombination, where you get exchange between two DNAs that share a similar region of DNA sequence. So they'll be able to basically exchange between themselves. They'll base pair in the similar regions so that you can get exchange that way. Site-specific recombinations where you get exchange at only a particular site, and then there's transposition. You remember those jumping genes? Short DNAs that can move from one chromosome to another, such as transposons, right? 3 functions of homologous recombination include assists in DNA repair, links sister chromosomes to properly segregate them between self and daughter cells during, mitosis and meiosis, and the source of DNA exchange and therefore increases genetic diversity. So, homologous recombination in bacteria. Replication fork encounters damage in the template strand. The replication fork will collapse due to the creation of a double-stranded break, and it leaves 2 parts of the strands to be processed, right? The 5' ending strand is degraded, and the 3' single-stranded strand has extension is bound up by recombinase pairs with complementary sequences. Intact duplex DNA can invade the duplex, creates a branch structure of 3 strands. The branch moves to create an X-like structure known as this holiday junction or holiday intermediate. And a nuclease and ligase can restore the structure of the replication fork. So you get double-stranded break. Here you have a DNA nick here, replication fork will collapse, and you'll have this with a blunted end, and then this end, and they can come in. You can get strand invasion from this end to this, because they're homologous basically. You get strand invasion. Once you get strand invasion, this branch will migrate, and you can form this holiday junction. After branch migration, holiday intermediate resolution and ligation will occur, and you can continue on. And there's a better movie showing you the holiday junction here than that figure. If it works. Oh, maybe I can. Is it up there? Did I just disappear it? Oh, okay. It's working now. It's really slow today.
To use the holiday model for recombination, draw 2 duplex DNAs which undergo cross-strand exchange. Now branch migration occurs, forming a heteroduplex. Bend both duplexes away from the crossover at a 90-degree angle. Next, rotate the lower half of this structure 180 degrees to uncross the crossover. The holiday junction can be cleaved either horizontally or vertically. Horizontal cleavage gives non-recombinant heteroduplex products. Vertical cleavage gives recombinant products which are hybrid.
So that's a lot better than the last figure. So you get recombinant products. If you see here, this chromosome was exchanged, you exchanged the ends of the chromosome basically in these homologous chromosomes. And these ones you only just exchange just a small piece. Does that make sense? Let's see if I can get this to work again. So the REC-BCD nucleases, helicase processes the DNA ends, binds to the broken end and unwinds the hydrolyzes, and so forth, and occurs at this chi sequence. The chi sequence on the 3' end binds tightly to the REC-C recombination C. 1,000 chi sequences in E. coli binding REC-C chi halts the 3' end strand's movement through the complex. The 5' strand continues to be degraded. And this shows you that REC-BCD helicase nuclease complex with DNA there. And there's the figure from your book. So the Rec A promotes the previous process of homologous replication. Rec A recombinase becomes active via assembly of thousands of subunits. The subunits assemble cooperatively on the DNA in steps of nucleation and excision. So the Rec A basically acts similar to like a single-stranded binding protein that helps stabilize that single-stranded protein. This shows you a picture where you have single-stranded bound with Rec A, kind of stabilizes that sequence, or that single-stranded. So RUVAB, well, this is Rec A again. Filaments with 3 helical turns of DNA. I always like the pictures on these. The RUVAB complex promotes branch migration, where the 4 arms of the holiday junctions come together. Hexamers of the RUVB bind opposite arms, use ATP to propel that DNA outward. And then RUVC is the nuclease, binds the RUVAB complex and cleaves the holiday intermediate on the opposite sides. The DNA ligase then seals the nick again. And you end up with either like this on opposite sides, or you have the chromosomes completely shared, or you just have a small piece if it was cleaved this direction, right? So in eukaryotes, we see it occurs frequently in meiosis, right? You know, meiosis is where the 2 chromosome pairs separate, and those are homologous chromosomes, so it's a great time for recombination to occur. Initially produces 4 homologous chromosomes during meiosis, a tetrad, and the 2 pairs sister chromosomes are connected at the centromeres. The cell then divides twice, and the chromosomes are segregated into their daughter cells, like so, and you have the 4 haploid. So this is a time where you can get recombination when those pairs are copying and separating. So the recombination links sister chromatids together during the first meiotic division. In the second meiotic division, physical links created by cohesions align the chromatids and help them segregate properly. Crossing over, recombination occurs in the first meiotic division, and sequences called chiasmata are where the strands break and rejoining occurs. This process not only aligns the sister chromosomes for proper segregation, but it causes the exchange for genetic diversity also. And I guess the movie fits this model better. We have gene A and gene B. With homologous chromosomes, you get the double-stranded break in 1, strand invasion again. The invading 3' end is extended by DNA polymerase plus branch migration. Eventually, after the second and end captured event, the generated DNA molecule, two crossovers is then formed, branch structured called the holiday junction. Further DNA replication replaces the DNA missing from the site from the original double-stranded break. And then the nucleases can then produce the two different sets of recombinations, depending on which direction it cuts from, right? So homologous recombination and gene mapping. In general, frequency of homologous recombination between 2 points on a chromosome is proportional to the distance away and allows positions of genes to be determined via gene mapping. And this is how you can do that, how independent assortment of sister chromosomes contributes to that genetic diversity. So aneuploidy can lead to miscarriage or development disabilities, and that's when you have the wrong number of chromosomes, where they don't segregate properly. Improper alignment or recombination can result in gametes with no copies of a particular chromosomes and 2 copies and the other cell instead of one. Normal cells join with cells with 2 copies is trisomy. And then trisomy of chromosome 21, we've heard of that, is Down syndrome. That's the most common trisomy that occurs. Differences in male versus female meiosis explain the aneuploidy. In male cells, germline cells begin to undergo meiosis at puberty, and it is rapid and frequently occurs, whereas in females, it's a much longer term process with an extended suspended animation phase where the germline cells being to undergo meiosis in fetal stage, chromosomes align and undergo crossover, and then the process stops for 13 to 50 years until the egg cells mature. So it kind of sits there like this, crossed over from 13 to 50 years. Disruptions to the crossovers during this stage can produce that aneuploidy, and likelihood of damage increases with age. So, because you're likely going to have more disruptions the longer it sits there like that, right? So, it's limited to site-specific sequences. For site-specific recombination, uses recombinase enzymes with either a tyrosine or a serine. The recombinase makes covalent bonds with the DNA via the phosphotyrosine or serine bonds, right? Cleaves the strand, joins new partners, and exchanges reciprocal. And this shows you site-specific recombination that can occur. So you have in this case, you have a tyrosine that'll attack those strands. You end up with the cleavage, and you have phosphotyrosine intermediates where you have the strand that's covalently bound to the enzymes. You get rejoining on the opposite strands. Isomerization, you get this holiday intermediate, and then you get cleavage again, and then rejoining again, and you get recombination, basically, where you split those 2 homologous chromosomes. This is site-specific recombination, so these enzymes recognize specific sites to do this. And this shows you an image of the recombinase bound to DNA sequence. So potential outcomes of site-specific recombination, you can have an inversion or a deletion, which will occur if the recombination sites are on the same DNA. Intermolecular recombination occurs if the recombination sites are at different DNAs, and you can have insertion if you have the DNA circular, it can insert like a new gene. And this shows you pictures of how you would get that. So inversion, you just flip this sequence around and made it inverted, which can be bad if you have like a promoter here. It'll read that DNA sequence in reverse and be something completely different. Deletion and insertion, you can delete this and insert it into a different chromosome where you deleted a whole sequence. And recombination repair of circular DNA can yield a dimeric genome. Resolution of holiday junctions can create dimeric DNAs. And their special site-specific recombination system converts the dimers back to monomers. And this is how the dimeric genome is created and resolved. So you have the fork undergoing recombinational DNA repair. Termination of replication. Eventually you get this dimeric genome where you have basically 2 copies of the genome in one large molecule. Resolution to monomers by XERCD system, just cleaves those and recombines it. And then we have transposable elements, or transposons, are certain DNA segments that can change the relative position, move to a region or a new chromosome where there's specific sequences. And these carry genes for transposases, but some can contain extra genes such as antibiotic resistance and so forth. In bacteria, short 5 to 10 base pair sequence act as the binding sites for the transposases, and these sites become duplicated as it does this. So you have a transposon with these terminal repeats. And then the target DNA, the transposase, makes a staggered cut in the target DNA. The transposon's inserted. Then the empty sites get filled in. And you've obviously made a double copy of this site that parallels that transposon that was inserted. So replicative transposition is where the previous transposition cut and paste. Replicative is copy and paste. The entire transposon is then replicated. And this is how that one looks. So you have, we're comparing the direct transposition via replicative transposition. The free ends of transposition target the DNA after the cleavage. Gaps are filled on the left, or the entire transposon gets replicated on the right. And then you get co-integration again after the replication, site-specific recombination with the transposition, the transposon that was replicated. And that was basically it for the chapter then. A lot of DNA stuff here. Do you have any questions today?
In homologous recombination, like the crossing over or meiosis, is that directed to particular spots, or you end up chopping genes in half?
You can end up chopping, but I think like the site-specific one is directed, right? There's two different types you can have. There's site-specific recombination, right? And that's directed to particular spots. And then you can have the random one also. Where are we? So it'll both occur. But the site-specific is very specific to the sites that these enzymes will bind to. But they could be in two different chromosomes, right? So you could get, movement of genes from one to the other, which can have bad effects too, right? If you interrupt between the promoter sequence and some sequence that regulates the cell division, that's where you can introduce, something bad. But does that make sense? But it can randomly occur too, just where you have the damage.